All right, hello. It's Mr. Davis back with you again, and this is NCSSM Online. And what I'm doing now is I'm taking some images from your textbook and have created a presentation, and I'll kind of hit the highlights of what I think is important, explaining some of the key concepts from Chapter 1, the genetic code of genes and genomes. And this textbook is a little bit different than other genetics textbooks. Very often, genetics textbooks will take a historical approach. That is, they'll reveal the concepts to you in the order in which they were discovered. Well, as you know, there was quite a long time, like 50, actually 70 years between Mendel's discoveries and the elucidation of the structure of DNA. So in a lot of textbooks, you do a lot of work in classical genetics while sort of pretending that you don't really know what DNA is. But in this textbook, they go ahead and introduce the general nature of DNA in the first chapter so that you can be mindful of it as you're going back and studying what we call classical genetics, the genetics of chromosomes, Mendelian traits, etc. So the first topic that's usually introduced in genetics is the Griffiths experiment. For a while it was thought that there was perhaps some chemical, some molecule that perhaps held information, was influencing life. But the Griffiths experiment was really the first experiment to demonstrate how, or demonstrate that such a molecule could exist. Let's zoom in here and take a closer look at this experiment. Okay, the idea is that you can take pneumonia bacteria and inject that into mice. Now, the bacteria comes in different strains, sort of like different varieties, and when you inject the S strain into a mouse, the mouse contracts pneumonia and dies, and it's not a surprise that you get the S strain bacteria back out of the mouse. Now, there is a less virulent or more benign version of this bacteria called the R strain. And when you insert the R strain into mice, the mice remain healthy. Moreover, you can then isolate the R colonies or colonies of the R strain from the tissue of the mouse. Now, as you all know, you can kind of sterilize things by you can sterilize things by boiling them in water. You can sterilize water itself by boiling it so it makes it drinkable. And how this works is that you're essentially killing the bacteria and other microorganisms in the water. So if you heat kill the S cells, denaturing all the proteins inside them, um, what happens is, and then you inject those into the mouse remains healthy, and you don't isolate any living bacteria from the mice. So now, interestingly, if you take two things that are benign, the living R strain cells, which have not killed the bacteria, and the heat killed S cells, which have not killed the mice, excuse me, and then you mix them together. So you take two benign things and mix them together. You wouldn't expect that that should kill the mice, but it does. They contract pneumonia and they die, and strangely, you get R and S colonies isolated from the tissue of the dead mice. So now the question is, well, what exactly is going on here? I mean, what is it that caused, something deadly to kill the mouse? What is it that caused the S strain bacteria to be reborn again once it was killed? So Griffiths thought that maybe there was something in the S strain bacteria that was transforming the R strain bacteria turning it into S strain, turning it into the more virulent, nasty bacteria. So whatever this transforming principle is, has the ability to confer traits, to a different strain of bacteria. And so maybe this transforming principle is the genetic molecule. So there are some candidates out there for the genetic molecule. The DNA was known, but the structure wasn't known. It wasn't known that it was the genetic molecule. RNA was known, and proteins were starting to be understood. So we go to our next slide here. There were some more experiments that were done, and this is the experiment that ultimately proved that it was DNA that was the transforming substance. The idea here is that you can destroy the S strain bacteria with heat, but you can't destroy the transforming principle. You can't destroy with heat what moves out of perhaps the S strain bacteria into the R strain bacteria. Now, but you can destroy some of the activity of some of these substances by using various enzymes. There's a few enzymes that we can use here. We can take this extract from the S cells that still could transform R cells into S cells, and if we use RNase, we can destroy the RNA in that extract. Now, anything that has the ASE suffix on it is an enzyme. So when you add RNase to the extract, the S cell extract still has the ability to transform R cells into S cells. As you can see here, there are a few S cells left in the culture. Okay, so now you can also treat this S cell extract with, and we'll scroll around the image here, protease. Protease destroys proteins. Now, when you destroy proteins, that doesn't really destroy the activity of the S cells. The S cell extract. The S cell extract can still transform R cells into S cells. So the conclusion here is that neither RNA or protein is the transforming molecule. But if you use an enzyme that is called DNase, and as you might imagine, DNase destroys DNA, then the S cell extract, when cultured with R cells, no longer produces R strain colonies. So our conclusion here is that the transforming activity, the transforming molecule, is most likely DNA. So that was an important finding. Okay, now the experiment that really conclusively proved that DNA had the ability to transform cells, or I should say is the genetic molecule, is the Hershey-Chase experiment. Now the Hershey-Chase experiment uses bacteriophages. Bacteriophages phage is just a fancy way of saying a virus. These are viruses that infect bacteria. They insert genetic material into the bacteria, and then that produces more viruses, and they break out of the bacteria and move around, just like when viruses infect your own cells. Now, the question was, what is it that the viruses insert? Do they insert DNA or do they insert protein? Because those were the two candidates for the genetic molecule at the time. And actually, a lot of scientists thought early on that it was protein that was going to be the genetic molecule, because protein is a lot more complicated. As you know, there's 20 amino acids that make up proteins, but there are just 4 bases that make up DNA. So you can have a lot more complex protein molecules, and so the molecule that was the secret of life, the genetic molecule, was thought to be complex because life itself is complex. Anyway, what you can do is you can radioactively label the protein and the DNA in the virus. and that way you'll be able to see it readily. And you can radio label DNA with radioactive phosphorus, and you can radioactively label proteins with radioactive sulfur. So you grow your phages in radioactive phosphorus, and then you allow them to insert or you allow them to infect bacteria, and they insert something into the bacteria. Then you can throw the bacteria into a blender, and that knocks the phage sort of bodies off of the the bacterial cells, and then you can centrifuge, and ultimately you discover that the radioactivity is inside the bacterial cells. Okay, now when you're looking at phosphorus, when you're looking at sulfur, which is, remember, radioactively labeling the proteins, you allow the phages to infect the bacteria. You knock the phages off by throwing everything in a blender. But what happens is, and then when you centrifuge that, you find out that what you knocked off is still radioactive, but what was inside the bacteria is no longer. It isn't radioactive. And so the idea is that it is, so the conclusion here is that is what is being injected. Let me move over just a little bit. Well, what's being injected into the bacteria is DNA. So it's DNA that is holding the instructions for making more viruses. DNA is, and not protein, is the genetic molecule. That's the results of this experiment, a seminal experiment, the Hershey-Chase experiment.